we are going to talk about motion. And motion is a cue that is one of the very fundamental clues to the brain about the external world, and it is also a very a sort of common stimulus for many different brain areas are sensitive for detecting motion. So what is motion good for? Like, why should the visual system detect motion? Well, there are two folds. The answer is two folds. Two answers you can tell to that give to that question. One answer is the positive aspect of motion, like what are things that motion are useful for. For example, perceptual grouping is something that is highly dependent on motion. How you define an object in the world, of course, you can define an object with its color, with its edge, with lots of things like that. But perhaps the most prominent clue to an object is common motion. At least objects that matter. What objects matter most for living things? Other living things, other animated things that they might eat you, or they might be food for you. They might the visual system is very sensitive to that, right? So you have to be able to group parts of objects that move together. Here is another example. These are antique, like vintage videos made in 70s, but I kind of like them because they're really low tech and they communicate the point. So when you have a bunch of dots that move together, they start to induce a shape for you, because now your visual system can use that motion cue to reconstruct shape. And it is also very useful, motion, for detecting fine 3D structure of objects, even without stereo vision, without a stereopsis, without like using the two eyes. Like, even if you use one eye and look at that pattern, when it moves, you can see the structure in it, because your visual system is trained to basically calculate what kind of real object would give you that projection when it moves. And the other thing about motion, which is also related to 3D vision, now that we are on the topic, is motion parallax, which is basically the perspective. It's just dynamic perspective. When you move, when your own head moves, the world moves on your retina, and the farther a point is from you, the less it moves on your retina. So that movement can be used to basically decipher the 3D structure or 3D layout of a scene. And actually, I want to use that particular point to mention my second issue about motion. Why motion? Why should the visual system detect motion? Well, the first answer was that motion is good for all these things that I mentioned. The second answer is that you got to get rid of a lot of motion on your retina. Your retina is exposed to motion all the time. You move your eyes 3 times in a second. So this idea of having a stable image on the retina and using that to sort of extract edges and reconstruct the image from that static image is just wrong. So if you are on the retina, if you're a like if you're a nano demon living on the retina, and you are like size of a photoreceptor, what do you see? What you see most of the time is that that photoreceptor is turning on and off, on and off, on and off, and on off, with regular patterns or irregular patterns, depending on what is happening out there. So how do you make sense of that? How do you make sense of what is out there in the world, by just having that dynamic information, that pixel turning on and off? Well, obviously, that pixel alone is not that useful. So you gotta look at the neighborhood. You gotta look at the, just like when you detect an edge and you need to see patterns of light and dark, how are they organized to make your little Mexican hat to distill the edge out of the visual junk that comes to the visual system. Here there is this idea of a motion detector, and almost all, not all actually, but almost all, and there are some interesting ideas out there, which is uh, I'll be happy to talk about them in the Q&A. But most of the mainstream models of motion detection based on this idea of having a series of lines that converge, and they basically, when you have a motion, when something moves, oops, sorry, uh screwed it up. So when something moves, if you have a delayed line, if you have a if you have a delayed line, let's say here, the signal is sent, is buffered somehow in a line, and when the other receptor sees that moving stimulus, because you have a delayed line on the left branch, now the two signals arrive at the same time and get summed up and create a motion, triggers a motion signal. Well, how is that delay implemented in the visual system is an interesting topic. It varies in different parts of the visual system, and it is still an ongoing debate. How is it exactly implemented? But you basically need delayed lines to be able to encode direction of motion. This idea is called a Reichardt detector. That's how we know it, and there are multiple variations of that. So now, if you have local neighborhoods, we are still on the retina. You have local neighborhoods, and you have managed to uh, gather signal from a local neighborhood on the retina and put some delay line on some of the pathway. Now you have motion detectors. Now you can detect edges moving in different parts of the retina. Uh, are you done? You're far, far from done. First of all, you have to know, is it the kind of motion that you want to throw out, or is it the kind of motion that you want to keep? The kind of motion that you want to throw out is usually uniform, right? Because if I move my eyes and that induces motion on my retina, that's a uniform motion. Well, if I am moving my eyes smoothly, if I am moving my eyes with saccadic eye movements, like ballistic movements, I leave one point and land another point, I have the opportunity of just just getting rid of that whole event that is coming from the retina to the brain during a saccadic movement. Although that is also a debated topic, but I kind of wanted to do this experiment together, but I thought that's too lame to do it all of us together. But I recommend you to do it with yourself and a friend of yours. um, get your friends to look at a mirror and move his eyes or her eyes, and just look at the eye movements in the mirror, small eye, short range eye movements. You'll see them easily. Then get the mirror in front of yourself and look at the mirror and move your eyes just slightly, not too far. You don't see your own eye movements. There is no way you can see your own eye movements, because that movement is censored sort of by your brain. That is a kind of movement that you don't want, and you sort of you suppress that. And there is some literature about that. It's called saccadic suppression. But anyway, what you want to do at the end of the day with the patterns of light that are emerging on your retina is to make sense of them. And to make sense of them does not just mean that, I mean, this is sort of a physicist's approach to motion, that for a physicist, motion is a vector, right? It has a direction and a size, like speed and direction of motion. But for biological things, that's not motion. That's the building block of motion. motion is very informative. As we talked about the 3D structure, objecthood, a lot of things can be extracted later on from the motion to actually make sense of it. Make sense of even the semantics of it is interesting, right? So a dynamic pattern that emerges on your retina has some objecthood, object information with it, has semantics with it. So motion is something that the visual system is stuck with throughout. You have high level motion, low level motion, and in some sense, similar to the case of color, we know a lot about low level motion processing, but we don't know much about high level motion processing. That's sort of a similar case because it's harder as you go to the higher levels of processing. Another thing that I want to communicate is that motion is like other things in vision. You remember we talked about the fact that your brain is actively creating a model of the world in your head, like how things are, and testing that with the incoming stimulus, right? So what you perceive is an active is an active process, is an active percept that is generated by your brain. Here is an example. This is called a motion quartet. So if you go, how many people see that as vertical motion? Let's see. I see. All right. Good. Good. Fair amount of people. How many see that as horizontal motion? Aha. Okay. So they are vertical. Now do this. Just cover the bottom row with your hand. Now remove your hand. Now they are moving like that, right? And you might get stuck in that percept. At some point it might switch back to that. There is no preference for them, right? So if you wanted to switch to vertical, cover one of the columns and then remove your hand and it switches to vertical. If you want to switch it to horizontal, cover one of the rows and then remove your hand, and then you're stuck with that explanation of this incoming stimulus. And if you are a sage, if you are like a Jedi, master Jedi, you can do other things with it too. Like you can rotate it. You can see if anybody can do that. You can imagine it as a rotating, constantly rotating clockwise. It's hard to induce the percept. I kind of, when I do this with my fingers at the right speed, I suddenly switch that to that perception that it's sort of a rotate. Did anybody get that?
I can change the percept by changing my eye movements. So I could do the rotating one by making my eyes follow.
Interesting. Interesting. That's right. So follow where it should be, where it's supposed to be.
Hmm.
That's a good trick. That's a good trick. And now, can you rotate it counterclockwise? The same thing, right? So this is an example that these motion motifs exist in your head. You just apply them to the incoming stimulus, and your brain is constantly calculating, is it this, is it that, where is the cue to tip the balance in favor of one of the ways you can understand the world. But the way you understand the world is made up here in your brain. So,
Arash, can I ask a quick
uh-huh,
question?
please.
So this was actually from something
Who
you
is,
said before. This is Sophie,
uh-huh,
sorry.
Sophie, Sophie.
Yes. Before you
Mhm.
started on this stimulus, I was just wondering, I have a better sense of organization from lower level to higher level static image processing, but what
Mhm.
does low level motion to high level motion, what does the trajectory of that look like? So where, for example, does perceiving an object from its motion versus the
Mhm.
motion parallax kind of phenomena
Mhm.
lie in that? Is it a line, like a linear kind of progression, or?
You mean in the brain?
Yeah.
Yeah, we'll talk about that. Actually, this whole session is about that. That like
Okay.
how are these different? So hold on to your question and then bring it back
Okay,
as
sure.
we move forward.
Mhm.
Um, so so The other thing that I want to communicate before we dive into the mechanistic discussion of how these things happen is that again, what you perceive as motion is made up, is a predictive model of how the world should work. Here is another example of that. Uh, what is the delay of your neurons, let's say in your cortex, to respond to a moving stimulus? It's something in the order of 50 to 100 milliseconds delay, 50 at best. 50 milliseconds is a long time when motion is considered. If you are an animal, you are in a hunting scene. Some whatever tiger is about to jump on you, and you have to just dodge. You have to dodge the attack of an animal, or a rock is thrown at you, or you want to catch an animal or something like that. You have to move really fast. In 50 milliseconds, a lot happens in the world. And if you are that lagged behind, you're going to die. So motion, this is an interesting property of motion. You would think that motion, because motion by definition is delta position over time, well, over past time, you would think motion is about the past. About you to calculate motion at every second, you need to have at least a little bit of history behind that, right? So motion, in a sense, is about the history of a stimulus as the stimulus flows into the visual system. However, the visual system, I mean, what is good about history? History is useless if you don't make any predictions about future with it. So that's what the visual system does. The visual system uses the motion to interpolate where the object will be. Uh, Otherwise, motion is useless. I am behind the actions in the world and I will go extinct. So this is an example of that, this effect, this phenomenon. There is a huge literature on it. It's called flash lag effect, or motion interpolation, in a sense, in a more general sense. So as you see, if you fixate on that dot at the center, uh, every time that the two vertical meridians of these two circles are exactly parallel, they flash red. But you don't perceive the flashed red bars as parallel, because your brain, even the two horizontal bars that are about close to the fixation point, when the flash happens, they don't you don't perceive them perfectly aligned. They are a little ahead of the dots. Although physically, when the bar is flashing, they are exactly aligned in the cardinal directions, right? You don't do that because motion is about future. Motion is not about the past, and that's a quite important thing to keep in mind about motion. How does that happen? Well, a lot of mechanisms go into that, and that happens in multiple different stages in the visual system. So now we start addressing Sophie's question. When does that start? Well, it depends on what animal we are talking about. Motion is a very old, important stimulus. If we are in, let's say, if we are in frogs, salamanders, even even birds, I believe, I guess, have that. Mammalians, in mammalians, it's a little trickier. Non-human primates definitely don't do that. But check this one. This is an example. Hmm, I shut up.
Is there supposed to be audio with this video?
Oh, you don't hear that? Oh, the
I
audio is not coming.
No, yeah, I can't hear it.
Ouch. Okay, doesn't matter. Okay, I'll do the audio. I'll do the audio for you. It's a frog. It's actually one of those old footages that I found. This is a vintage footage. I like them because they expose you. I mean, they show more. Look at that. There is taking out the vitrae of the frog eye and putting an electrode in a ganglion cell. And now he shows that a ganglion cell in frog retina is responding to motion, a particular direction of motion. And here is the interesting part. If you move that little thing over the pattern in a specific direction, the cell fires. If you move that thing with the background, the cell does not fire. That's what the sound is, the soundtrack is. So the cell is silenced there, but when you move it with respect to the background, the cell goes crazy. That's an example that shows even in the retina, even in the frog retina, you can detect, first, you can detect motion. Second, it's a specific motion. It's just directional. It's not motion in any direction. And third, and most importantly, this is motion with respect to the background, not absolute motion. This is relative motion. And that is the motion that you care about if you're a frog, because you want to catch that fly that is flying over. And so here is an example of a paper which I highly recommend. It's a very interesting paper by Marcus Meister's group. And the idea is that when you go record from retinal ganglion cells that are motion selective, and this is in done in salamander and in rabbits. So the plots to the left are salamander, plots to the right are rabbit. And as far as I know, we don't have any evidence of such things existing in the non-human primate and human brain. So this is not a primate thing, but you see, even rabbits have that. Uh, and it's not that we have explored the non-human primate retina perfectly. It's things are complicated there, so that might exist there too. So what is the finding? The finding is that if you take a position on the retina that is the center of the receptive field of the ganglion we are recording from, and you flash a bar in there, just the brief flashing of a bar with some delay. So do you see my cursor? So this is time zero. That's the time that the flash happened. With some delay, well, it's a neural structure. So with some delay, it responds to the flash and dies off. The response dies off. And you see the same phenomenon with slightly different lags in rabbit and in salamander. Now, that's a bar flashed at the center of the receptive field. Now you take the bar out there and move it across. Just move it across the visual field. And it crosses that receptive field center. And what you see is that, let's say, take this plot. When the bar is out, the bar has not arrived. This is the time that the moving bar, arrives the center of the receptive field. Uh, Way before that, the neuron is firing. This is the firing rate. Way before the bar arrives, and even after it arrived. And it's interesting, it's asymmetrical, right? When it arrives, it sort of dies off because now the future cells should be firing. Here is another way of plotting the same data, which is quite fascinating. So imagine now we are looking at the population activity of these ganglion cells on the retina. Your x-axis is position on the retina. 0 is the center of receptive field, and negative and positive are corresponding to the other spots on the retina that are on the path of the moving bar, right?
Arash, can I
And
ask a
then,
question? Can I ask a question just so I
yes,
fully
yes,
understand?
Rosa.
So when you say like that dashed line represents the center of the receptive field,
Mm-hmm.
the area to the left of it If you were
Mhm.
to just flash a bar outside of the center, would you get a response? That is the receptive field large and the dashed line is the center, or is it that it is the neighboring cells that are
Beautiful question. Beautiful question. Look at this. The red bar is what you described. The red bar is when you flash the bar here. Oops. Flash the bar. There is no moving stimulus. There is just a flashing bar. At the center, you get the biggest response. You go out, the response goes out, and it sort of dies off. So the neuron is wired to those positions. We know that. It's just a wide receptive field, actually. However, when you move the bar, this is what happens. When you instead of using a flashing bar, when you have a moving bar, you see this wave of activity ahead of the bar
Mhm.
on the retina. So you see that is getting distorted to one side, and this is into the other side, depending on if you move the bar to the right, to the left, or to the right. The population response of retinal ganglions shift to the left or to the right. Although if you don't have any moving bar, it's a symmetric response, right? So it's a receptive field. You go out of it, this response dies off. But when you move something across it, then the activity pattern changes. The neurons that are ahead of the traveling wave, they fire up faster. And that's amazing. That's amazing to have that in the retina. This is motion interpolation, predicting motion, basically, that is happening at the retinal level, which shows how important that is. And of course, these functions are, I mean, we don't know much about the non-human primate eye. We have not found these in the non-human primate yet. They might be there, or we might have missed them, or maybe this function happens higher up in the visual system, but it definitely happens.
Arash,
Fernando,
why do you refer to this as predict?
because at this time, let's say at this, well, this is a position, right? Let's do the time plot. Oh man, here we go. So because at this time, right, when the bar is moving here, this is time, right? When this is firing, the bar has not arrived there.
It's anticipating.
It's anticipatory response. Like if you look at the delay of these neurons, the neuron is delayed with respect to the stimulus. So when something happens, there is a delay and there is a response. But when that something that is happening is a continuous motion, the response builds up faster. The
Yeah.
response actually builds up in anticipation of the arrival of.
Okay. Yeah. Maybe I have to think a bit further, but isn't it possible that this cell is wired to other cells? So there's actually connectivity there, and there's other cells that are being activated.
Of
So
course,
so
of course,
it's just like a consequence of the wiring. The cell is not
of
predicting anything.
course.
It's just
Of
summating.
course, of course. The cell is just a cell. It's a bag of ions. It's a leaky bag of ions. It cannot decide anything or predict anything. I should. So when I use that language, of course, I don't mean anthropomorphic language. Yeah, it's wiring. It's the way the wiring works, and the wiring is pretty amazing. There is more and more evidence are coming out of this. A lot of wiring happens in the bipolar cells of the retina that are connecting the photoreceptors to the ganglions. And a lot of this processing even happens without involvement of the cell body. It's dendritic computation. So that's a very fascinating topic, how the system is wired to do that. But of course, it's a wiring diagram that causes that. It's the way it is wired along a receptive field that causes this anticipatory response of the receptive field. Yeah, the wiring is basically how the prediction is implemented, if you will. And yeah, of course, the cell, it's just a simple circuit. So now, although it's not that simple of a circuit, it has it. I will sort of go on a tangent that will sort of take a whole day to talk about that, and I have to stop myself. But the motion detection and the local processing circuits in the retina are so fascinating, and we still don't know a lot of things about them. They adapt to the dynamics of the stimulus. They adapt to light. It's a very interesting circuit. But sure, it's a circuit.
Arash, a quick question: Is velocity
Tim.
encoded anywhere in this signal? Like how fast?
Very, very, very good question. Very good question. In general, when you record from lower levels of the visual system, most of the responses are tuned to temporal rate. You remember that dot I showed? The dot was turning on and off. When you have a moving stimulus on the retina, from the point of view of a local sensor, things are just turning on and off, right? There is no movement. And these early neurons of visual system are mostly tuned to temporal rate. Some of them respond to faster temporal rates, some of them to slower temporal rates. However, as you go higher and higher, you get more and more tuning to speed. How do you calculate the speed? You cannot calculate the speed with one sensor. You need more than one sensor to do that. And as you go deeper into the system, you get more and more speed selectivity compared to temporal frequency selectivity.
But with the anticipatory curve, right? If it was a faster movement, then you'd have a smaller, you'd have less time of firing before the stimulus got there, right?
Right.
So
Yeah.
wouldn't that have some kind of speed information in it?
That's a very interesting question. That if you're asking, if I understand correctly, you're asking now, if you vary the speed of the anticipatory stimulus, would the anticipatory response be different?
Mm-hmm.
Right.
Yeah.
As far as I know, the answer is yes. But I should dig into papers to show you the actual papers that show that. But as far as I know, the answer is yes. Actually, that might be in that paper that I showed. I'll check and find it for you offline.
Okay, thank you.
So now we go to V1, area of V1 in primate brain. This is we don't have sound, right? How do I fix that problem? Any advice?
Is it because you're listening through your headphones?
Oh, that might
so
be.
If you turn your headphones off and let your speaker play, it should come through the microphone.
Right. Okay, so never mind it. Okay, I'll just play the soundtrack for you. So again, this is another example of an early visual cortex neuron, which is David Hubel and Torsten Wiesel doing their experiments in V1 again, and he's moving that bar of light and determining the boundaries of the receptive field. And then he shows that within that receptive field, when he moves the bar only in one direction and not in the other direction, the cells fire. The cells very specifically fire, and the direction that they like is that direction that he just drew. So this is different from orientation selectivity. Orientation selectivity was that you wanted an oriented line to respond to. And you have this, as a V1 cell, you have this best direction that is driving the cell most. Now, usually when you find the best driver, orthogonal to that, one direction or the other, if you are a direction selective cell, and a lot of cells in V1 are, that will drive the cell. So basically, the orientation sets the edge direction, and then the motion direction is always orthogonal to that, which drives the cell most. And you can actually encode motion in V1 cells in early primary visual cortex.
Hey, Arash, quick question.
Please, who is speaking?
It's me. It's Nahom.
Nahom, please.
First of all, I think you can. The audio issues you're having is probably because when you shared it, you didn't include the audio, so you can reshare the page, and then it will give.
Oh, let's try.
Before
Let's
you click share, there's at the bottom there's sound optimization or audio.
screen share. Share computer sound.
Yep.
Optimize screen share for video clip. Aha, let's see. Thank you. Let's see if it works. You see, it's very robust. It's not something you detect with a like small p-value. You hear it. Now he's being playful. All right. So so now if you are in V1 cortex.
Sorry, Arash, is it? I just want to know: is it always unidirectional, or is it sometimes like does it sometimes go in both directions?
Yeah, some of them are not direction tuned. Basically, those ones, if you move them on either direction, you will get some increase in firing rate because you just have more motion energy in the scene. But the ones that are called direction selective, they are unidirectional. And they're usually suppressed by the opposite direction.
Oh, okay.
One
All right,
question
that's very
before
interesting.
we continue.
Mm-hmm.
The previous slide you were showing.
Uh-huh.
So on the left side, the plot with the dark straight line and then the dashed line behind, well, around it. So what do they represent? The dashed lines.
So the dashed line is basically what he drew here. Is the receptive field? It's basically his measurement of the receptive field. The two side lines are when the stimulus goes outside the receptive field, and the top and bottom ones also are the one when the stimulus goes outside the receptive field. So that's roughly, although it raises the question of what is a receptive field, right? And how do you measure a receptive field? This is sort of a hand measured version of a receptive field. I believe you can ignore them, and you will still be fine. The fact stands. Even if you imagine a circular receptive field, there doesn't change anything.
Okay, thank you.
Sure. So now one problem is with motion processing in low-level vision in general is this problem that is called aperture problem. And it's particularly pronounced for V1 cells. V1 cell is sort of the biggest first filter of motion in the cortex. And each V1 cell is looking at the world through a small aperture. Well, it's not as small as the retina. It's not just one photoreceptor. It's about 1 degree in size. So like sort of, I don't know, like half of the size of your thumb from one arm distance, 1/3 of that. That's the size of the V1 receptive field. And you see the world through that hole, through that aperture. And you are a filter, you are an orientation detection filter. You're a Gabor filter, right? So whatever that comes through, you will see through the lens of a Gabor filter, right? So you see the value of it is the orientation energy in it. Now, the thing is moving and you detect the motion in it, right? You detect the motion, and the motion is actually orthogonal to the direction of the orientations. Fine. So if there is a zebra out there behind that aperture, where is the zebra going? The zebra is going there. However, in reality, that's a problem with apertures. Like when you filter things through a linear filter, you lose direction. When if a zebra is moving from left to right, you would perceive the zebra as going slightly up, because your motion filter is detecting basically the motion that is orthogonal to your orientation signals, and that's a problem. That's a problem with motion. And there is a lot of little sort of tricks that the system does to get rid of that problem. Like if you have sensitivity to this corner, the corners solve that problem. However, we are not completely getting rid of that. And remnants of that remain in our perception. And that is the interesting part, because evolution solves the problems as much as they are needed, as much as we can survive. Do you remember this illusion I showed you a few sessions ago? Did anybody try to explain this? So if you do that, right, if you fixate at the center and move your head back and forth, you see the rings rotating. Does everybody see that? Yeah. So why rotation? Why are they rotating? So let's take one of these little things. That thing has an orientation signal in it. It activates a V1 cell. Let's say you have a V1 cell that has an excitatory lobe and inhibitory lobe, like a really simple Gabor, oriented that way. This stimulus has a contrast and activates that V1 cell with that orientation. Now, when you move your head close, what is happening on your retina? On your retina, this image is growing as you move your head close. So what is happening is that this patch is moving out, right? Is moving into this direction when you move your head close, because the whole thing is expanding on your retina. When that moves into that direction, the V1 cells that are responding to this detect this motion because of aperture problem. Because V1 understands motion only orthogonal to the direction of its preferred direction. And all right, so the actual physical motion is a little now tilted, distorted, because of the way your system is wired. And that happens for all of them. And that when you sum those vectors, you get that very nice rotating signal, and you can actually interlace them, have them rotating in these different directions, and they still rotate for me, actually, even with that clutter. So that's an example of how the system has to solve the problem of direction, of orientation and direction. And this is the way the system has solved it, and it comes with a toll that we pay, right? And that's fine. Basically, we didn't really fix that problem, and it gets worse. It gets worse. So now, why are those guys moving? Can somebody explain? Malcolm.
Is it because like you're looking around with your eyes. Like if you hold your eyes still at one spot, it stops.
Excellent observation. Yeah, fix your eye at one point, the whole thing stops. As soon as you start moving your eyes, things start moving around. It actually shows how much your eyes move and you don't notice,
Yeah,
right?
that's
Because
crazy.
your brain again is sort of is censoring all those things. You don't notice that. So basically, your eye movements cause that pattern to move, and because of aperture problem and related wiring issues, you start seeing motion in a pattern like that. Okay, another topic that we have to cover, it's impossible to talk about motion and not talk about adaptation to motion. You've seen it, but it's fun. Let's try it one more time together. Fixate at the blue dot. So as you keep your eye fixated at the blue dot, what is happening is that, and I want to create a strong after effect. So keep looking there, fixating there. So you must begin to see Buddha breathing slowly as the adaptation build up. So when we go into the adapting phase, there's this inward motion, and your neurons that respond to that direction of motion are getting tired. They keep firing, and they run out of mediators, right? Neural fatigue. And now when you see the stationary Buddha, the neurons that are encoding the opposite direction fire more because they have been suppressed. Is the Buddha beginning to breathe for you? Keep your eyes there. Okay, now I want you to do something really fun and look at your palm. Or the wall or whatever. So what happens is that now you're, well, it's sort of, it's there for half a minute or so, and then it sort of disappears. So what is happening here? Adaptation shows you that there is a structure in your brain that selectively responds to motion, and you can adapt those structures out. And this has been a very valuable tool in studying how motion is decoded or encoded in the brain. Uh, one thing that now that we are here, I want to mention, and this is a general point. I'm using motion adaptation as an opportunity to bring this up, but this is a really general discussion, is that when you have a phenomenon like that, you look at that motion signal and you get adapted to it. And then if it is a contracting motion, then things look expanding to you, right? If it is a left to right motion, things look like moving from right to left after you are adapted to that. So the way I presented it for you was that this is because we are wired not with silicon chips. My eye is not like my cell phone camera that is a silicon chip solidly responding to light no matter how long you expose it to light. It's my eyes are a biological devices. They run out of chemical mediators, they run out of juice basically. And that causes adaptation. When you start bashing over and over on same neuron, it gets tired and you get these adaptation or after effects, like the color after effect, like the motion after effect. Or is it the case? Or is it the case? Another interpretation of adaptation, which is very important, is that it actually has a function, and it has a very important function. And the function is that if you are living in a world that is constantly expanding, for example, you live at the tip of a rocket, right, that is moving forwards, right? There is expanding. You should get rid of that. You should get rid of that motion. You should not see that motion. That's the norm in your world. You remember information theory? Things that are more common are less informative. You got to get rid of them. And that's what adaptation does for you. In that sense, that's a and I think that's an important epiphany about the mechanisms of the brain. If you want to make a robot, so here is the challenge for you. If you want to make a robot, you're not bound to the crappy biological material, fat and water and stuff like that. You have silicon chips, gold wires, reliable system, electronics. Do you implement an adaptation function in your robot or not? Does your robot have After Effects or not? What is the ideal robot?
Arash, it's a very complicated question. And
Uh-huh.
first of all, the reasoning from information theory that if you live in the tip of a rocket, that you should kind of
Uh-huh.
get rid of that, you could argue
Uh-huh.
exactly the opposite way.
Mm.
You could argue that you should become much more sensitive to variation about that state.
Beautiful. And to become more sensitive about variations of that state, the average of that state should be nulled out. So basically, you want your neurons not to be at the saturation point that are, wow, expanding motion, fire, 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 and then you will miss the nuances in there. If you get adapted to all of that, now expansion, expansion, you're calm. Your neurons are not firing. A little bit of difference in that will make them fire. The,
But
the,
then,
the, the,
but
mhm.
but they're not fatigued. That's the point. The point I'm trying to say that the idea that it's fatigued is like would be a very misleading metaphor.
Exactly, exactly. So the thing is that so there is literature around that, and there is debate that is neural adaptation fatigue, or is it normalization? You're renormalizing your world to the new realities of the world. Well, the answer is both, because it's a crappy biological system. It got fatigued anyway, right? And then the wiring of the system is taking advantage of that, of the system. The system has adapted itself to adaptation as a byproduct of how this system works. And if you want to make a robot that functions the way we do, you have to implement that fine. Your robot should actually get adapted. Otherwise, the robot will not have the good dynamic range for seeing the differences between others. And that is true about everything, right? That is true about faces. You go to a new country, all the faces look, wow, so different for you that your face neurons are going crazy. They're firing like all like, oh, that race, whatever race that country is, right? And all of them look the same to you because that's such a big stimulus that you are out of the dynamic range for seeing the differences. But then you live there, you adapt there. You know what? Actually, this is another deep intuition about adaptation. And I just say it and move away. I have to shut up. But adaptation and learning are same things, same direction. Adaptation is fast learning. Learning is a slow adaptation. Uh, Basically, you're adapting yourself to the world, to the shape of the world. You're learning the world. In information theoretic sense, you are adjusting your bits of information. How many bits should I put for that motion? How many bits should I put for this color? And that economy is adjusted by how common things are. And adaptation is basically a function that helps you normalize the stimulus. I just wanted to bring that up to sort of, when you think about adaptation, it's possible to think about it as an epiphenomenon of how the system is made. But also it's possible to imagine that it is actually serving a function for the system. Another thing about the motion system is that it turns out that the motion system is more or less colorblind, more or less colorblind. Look at that video. Do you see the yellow moving? The yellow is moving with the speed of the blue. It's actually moving. Like if you, so when it switches to blue and black, you have luminance cues, right? The blue and black have different luminances. Even if you look at it through a black and white screen, you would see that motion. But the yellow one is sort of equiluminant, but saturated yellow and more whitish yellow, less saturated yellow. And so basically those lines are defined only with color. And it turns out that the motion system is not that sensitive to color. Well, it's not totally colorblind. It sees color, but it's surprisingly less color sensitive. Why? Again, that's a byproduct of how the system is wired. Most of the motion signal comes from the periphery of the retina. The periphery of the retina is where you have the maximum area, right? And that's easiest to calculate motion over. Most of our motion system is fed by the periphery of our retina, not by the center of the retina. Center of the retina is where you have the color cells, where you have the high density color photoreceptors and cones. So now let's move on from V1 to the bigger picture of cortical processing. There are other motion circuits that I did not mention at all. Like there is this ancient motion circuit in the superior colliculus, in the brain stem, which is what we share in common with our snake ancestors, our lizard ancestors, basically. And those are also motion selective. If something moves here and you turn your eyes there quickly, that reflect is happening in the brain stem, and we don't have time to talk much about that. But as far as the cortical processing of color of motion is concerned, there are two main pathways of processing in vision in general: dorsal pathway and ventral pathway. Traditionally, if you look at the traditional literature of motion, dorsal stream is where motion happens, and ventral stream is kind of motion blind, does not see the motion. That's the standard textbook version. It doesn't seem to be the case, however. But let's talk a little about the most prominent dorsal stream center for processing motion. And that is area middle temporal, area MT stands for middle temporal, or in macaques we call it area V5, which is the equivalent of the area in humans. And if you look at it, it's sort of here, it's here in your head. In the macaque, it's kind of a similar position. There you find neurons that respond to motion direction better than V1, more accurate than V1. Back to Tim's question, a lot of V1 motion cells are sensitive to temporal rate, like how fast something turns on and off. When you go to MT, it's speed. It's mostly speed. It's selective to speed, calculates the speed. MT is an interesting area from embryology point of view. When you look at the embryo and how the embryo develops, you see, like here, you see that when the cortex starts to form, two spots appear in the back of the brain, two spots of highly myelinated cortex that have these genes expressed, and these hormones are made by these cells, and the rest of the visual system is based on the gradients of those hormones, is built around those two centers. One is V1, one is MT. So from embryology point of view, MT is a primary visual cortex. That's very important. We are used to, I grew up with this idea that V1 is the primary visual cortex. There are two branches. Serially, they pass the signal down, and eventually it gets to V1, V5 in the dorsal stream. That's not true in terms of embryology. MT is a primary visual cortex. It has high amount of myelin in there. The neurons, they are fast neurons. It's a fast pathway. It is it is fed mostly from V1, but it has a separate link that goes directly from thalamus. Basically, signal goes from thalamus to MT, and we don't exactly know what it does. If you look at the animal phylogeny, you see this interesting thing. Like this is marmoset. That's V1. That is MT. That is area of V2. This is human. So look at that. The gray bars are the V2 to V1 ratio. How bigger V2 is relative to the size of V1. This one is MT to V1 ratio, the dark one. So as you see, V1 and V2 has grown as we go from marmoset to human, but MT has kind of stuck at the same size, relative size. MT has not grown. And that sort of shows that MT has a more prominent role in lower animals. And even, I mean, in terms of both evolution and embryology and development, MT is sort of a primary area, but then it sort of loses its importance in evolution. It is perhaps less important for our brain compared to the marmoset brain when it is. Fernando.
I mean, I don't want to be, evolution is such a topic that invites for kind of potential accusations of discussion. So I don't want to. But I did want to mention something. I mean, I presume that perception of motion and motion signals is fundamental for any kind of visual organism.
Exactly.
Wouldn't it be a more reasonable way to think about it that whatever role MT needs to play doesn't need to grow anymore, rather than it becomes less important?
Maybe, yeah, yeah.
Or is another possibility that you no longer have specialized dedicated tissue because motion information is carried by other higher order regions, right? That you have motion bound to other information.
Could be. It's actually a very good question, and I don't have the answer. I don't know the answer, and I don't know if the field knows the answer to that question. And in terms of like, is it the case that Homo sapiens brain has other structures to process motion, and that's that, or maybe Fernando's idea is right, that was enough, that MT is enough. But the point that I want to make by this evolutionary comparison is that MT is a little bit of a black sheep. Is a little different from other areas. Is an interesting case. MT is histologically different, is myelinated highly, so it's like V1 from point of view of histology. From the point of view of development, it's also like V1. It's a primary cortex that has changed later in development and in evolution.
And actually, this is a question I'm really kind of important. I've been sort of worried about, not in a bad way, in a good way, but if you look at MT in humans, it's like sort of more like in the ventral stream. While if you look at monkeys and macaques, it's like pretty dorsal.
It's pretty dorsal. Yeah.
So I've always been puzzled by this, because in humans it's neither medial nor and it's really kind of like it's going very ventral. I mean, it's not far from lateral occipital complex.
Right.
So I was wondering, what is this all about?
Well, I don't know. I don't really know. The distinction, though, in terms of the pathway is comparable. In terms of what feeds into MT in human and monkey, it's very comparable. Although its position is more ventral, but in terms of circuitry, if you look at the Van Essen diagram, Feleman and Van Essen diagram, it's pretty. Actually, Feleman and Van Essen is monkey. If you look at the equivalence of that in humans, it's similar. It's the circuitry is rather similar.
And ventral stream in humans is literally on the ventral surface.
Is actually even more ventral.
It's not that it's lateral. So we talk about the ventral pathway in the monkey, and that's on the lateral surface.
Right.
But that whole map, like what we think are the homologs,
turned inward
on the ventral surface. So the fact that MT remains on the lateral surface in the human it's not the equivalent in the monkey in terms of its location. It is more dorsal relative to the rest of sort of ventral visual pathway.
Right. That's true, and it's very interesting observation. But on the other hand, you also have the superior temporal sulcus, and MT is like above the superior temporal sulcus in monkeys, and in humans is like sort of below.
Right. So there's been this massive cortical expansion in the humans between the superior temporal sulcus and all the ventral visual regions that happened most likely because of our acquisition of language and social processing, social sort of skills that are processed by tissue in that region. So like the TPJ, the theory of mind area, all of that, the region between STS and ventral surface expanded like 20 to 30 fold since we separated from our last common ancestor with macaques, whereas V1 expanded maybe 1 to 2 fold. So there's been this wedge essentially into STS that expanded and pushed the ventral map on the bottom and left the rest of the stuff up here. So the STS is not actually a good marker
exactly
on its own for trying to understand the relative position of these things.
Yeah, but I understand that point too, and I think, but in the end, things end up far away from other things, and MT has been pushed to a location that does not have to do with the typical. MT was the prototypical dorsal area, movement, speed, magnocellular.
Right.
Well, right now it is end up pushed up to close to V4 and other areas that are quite different. So I agree with this theory of ventral migration of the brain areas. I mean, it makes a lot of sense. And I agree that there's expansion of STS, but I think that this is not a minor kind of rearrangement of the circuitry. It does not seem to me. I mean, maybe I have to read more about it.
But what about its relationship to parietal cortex? Because I sort of agree with Fernando. Like MT is very close to parietal in monkeys, and in humans, they're like very, very distinct, right? And we know that in humans, like there's a lot of parietal motion stuff going on that's very distinct and separate from MT, which is pretty close to like biological motion processing stuff in humans.
Right. Well, it actually gets even worse when you think about that MT is an area that induces a structure in the visual cortex. If you look at the development, like you have a little ball of neurons that is going to turn into a brain, right? How is it determined? Where should it be V2? Where should V3 be? Where should V4 be? Like those retinatopies. And it turns out that you get this gene expression in these are the genes that create these hormones that are made by some cells in that ball of neurons, and then it's excreted and it creates a gradient in the tissue, and that gradients determine this should become V1. If you are at the lower gradient level, it should be V2. If you are even lower, it should be V3. And you have two of these sources, two of these gradients. One is V1, one is MT, is as primary. And then those two gradients determine what areas go in between. The bigger the brain gets, the more areas you get in between. So basically that map of areas is way more expanded in humans compared to macaques. And the details of difference between those two, I don't know actually to what extent we understand that, because in humans we are limited in how to physiologically study it. However, there are many other similarities between the two regions that are the basis of the homology. There is the histology similarity, common input similarity, common function, and also the lesions lead to the same sort of similar sort of behavioral deficits in both cases. That's the foundation of the homology, but they are not the same, because it's this Homo sapiens brain and that is a macaque brain. It's a, like much smaller, 100 grams, 1,000 grams, 10 times a smaller brain.
Yeah.
One thing I wanted to add just before we move on was, so you have like the echinotopsia, super specific deficit from lesions to that area in the human. But so in my research, I did like a motion, like perceiving object categories from motion derived signals. And so they were only looking at motion information from moving dots, and they could understand object categories. And from decoding what object category they were looking at across the brain, it didn't seem to me to be localized to really specific areas, what neural activity was able to do that. Like even ventral regions, obviously there's some communication about structure that's happening there, but I'm just wondering how specific that function might really be.
It's another good question. Good question. Bear with me for 2 more slides. Because I mean, first of all, this is about motion blindness that you mentioned. This is again one of those old footages which we fixed the soundtrack, but it's not going to help much because the soundtrack on this one is Japanese. But let's listen. Let's look at it.
ドイツ人のこの女性は14年前に脳血栓で倒れてから買い物に出かけられない、テレビが見られないなど日常生活で困ったことが起きています。コップに水を注ぐこともその一つです。水面が上がってきたのが分からなかったのです。世界でも彼女しか報告がありません。てっちゃんのように片方の脳でも健全ならこういうことは起こりません。彼女にとって外出は恐怖です。最近やっと主治医のチール教授のおかげで街に出られるようになりました。
The subcompartment of her brain that handles visual motion was damaged. She had no trouble seeing cars, but she couldn't judge their speed, so crossing the street became an ordeal. She avoided parties because of the way people suddenly seem to appear in front of her. And how do you follow a conversation when you can't see the motion of someone's lips?
She finds it very annoying to talk to people as well, because she cannot do lip reading. She finds it hard to even understand. This is something that we understand these days when we talk to people with masks. You realize how much speech understanding is dependent on lip reading. So anyway, you go to MT, I gotta finish. You go to MT, you record from MT, just like V1, you find these columns of cells that are tuned to different orientations, to specific orientations. They are also sensitive to a stereoscopic depth, as you can imagine, because motion is highly involved in depth perception as well. And the study that I want to, like, this is the penultimate thing that I want to tell you today, is this is a study by Daniel Salzman and Newsom's group. It's a classic study, very important study that shaped systems neuroscience in many ways. They created these motion signals. This is called a random dot kinematogram. And it's a bunch of dots that are moving randomly, like doing a Brownian motion. Now you can make 50% of them going upward. The rest of them are random. You can make 100% of them going upward. Basically, you can tighter the motion signal in there by making a group of them correlated and moving in one direction. And the more correlated they become, the easier it is to see the motion for the monkey. So now you get the monkey to tell you, just look at this curve for the moment, ignore that. Look at the open circles. This is the motion signal. This is 0 signal, means fully random motion. And as you go to positive values, you get more motion signal in favor of a direction, which is the preferred direction of a cell in monkey MT. So let's say they are recording from a cell. I am the monkey, they're recording from my MT, and there is a cell that responds to this motion. So when you create a stimulus that has 100% of the stimuli of the dots moving that way, you are moving this way. When the stimulus is chance is random dots moving with no motion signal in there, you are 0. When it's the opposite motion, when you get the dots correlated moving the opposite direction, that's the negative direction. And you ask the monkey to tell me, was the dots moving in this direction or in that direction? You basically put 2 dots next to it, and the monkey is supposed to look at the dots that is aligned with the direction of motion and answer you by eye movements. So what you get is that the monkey is reasonable. The monkey is at 50-50 in the middle. The monkey gets better at reporting that direction when you increase the signal, and vice versa. When you stimulate MT cortex, when you stimulate those neurons, this whole psychometric function shifts to the left. What does that mean? It means that now at the 0 signal, the monkey is more likely to say, I see motion in that direction. And that is fascinating. That is fascinating. You can induce a direction of motion perceived by stimulating MT cells that are responding to that direction. And more importantly, if the receptive field of your cell is there, you show a stimuli over there, you don't get any shift in the psychometric curve. It only happens if you show the stimuli at the receptive field of that particular column of MT. So that shows that MT is actually causally involved in perception of motion. And now I want to end with the question that Sophie brought up. The question of pattern motions, or biological motion. Motion, as you remember, it's not just a movement in linear direction. Motion has a lot of signal in it. One of the most famous ones is, do you see my screen now? No. Okay, I gotta share again. Let's go here. Okay, now do you see that? So that's a biological motion. That's a human body moving. We don't see any structure in there. We don't see the face. We don't see anything. But we can say that's a human body moving, because the motion is meaningful for the system. We can extract that information and make sense of that. And it's more sophisticated than just there is a human moving here. Like if you follow the statistics of natural humans, now it's a male moving, and now it's a female moving. So the type of movement, even the anatomy of the body determines the shape of the movement, and the visual system picks on those cues, right? There's a lot you can decode from that. Like, that's a heavy person moving, it's a light person moving, and you can actually parametrically vary these and study them. And I believe that's what Sophie is doing. And what you learn is that the ventral stream is also selective to motion, at least what we know from physiology. This is an interesting work. This is a very interesting work. Recordings from superior temporal sulcus, the upper bank of superior temporal sulcus. STS is where you find the face neurons on the bottom of it. And this is a work from Gazanfar et al. He was at Max Planck at the time. He's at Princeton now. He found that there are neurons in upper bank of STS that respond selectively to monkey calls. You know, monkeys have these 4 different specific types of calls. And one of them is this coo they make. It's like, ooh, ooh, a sound like that. And the other one is this grunting sounds like, sounds like that. And you find neurons that are responding to this sound or that sound. Now, here is the interesting fact. If you play the video of a monkey's lips moving and making, like when they do the coo sound, their lips move like, the lips sort of round up and get small. But when they do the grunt sound, their lips sort of open up and they have a different mouth movement. So if you take a circle and make the circle, basically adjust a circle into the inside of a mouth as the monkey makes that body movement, that lip movement. A circle down here, the disc, changed shape in a very specific way, right? That's a circle that is inside the mouth, gets bigger, 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 smaller, and it sort of moves up and moves down with the lips opening up and moving up and going down. It's a very specific dynamic. A disc getting large, moving up, getting small, going down. For the grunting sound, the shape of that disc would be totally different because the shape of the mouth is different. And he found that if he only plays the dynamics of that disc, the shape of the mouth, basically, the very abstract shape of the mouth, to the neurons that respond to that coo sound, the same neuron will fire. That shows that an aspect of biological motion, which is lip reading, the way the lips move, are encoded directly in the cell that is responding to the sound relevant to that. And that shows that, okay, we have been studying motion in the context of linear lines and whatever, Gabor filters. It turns out that motion is being used for decoding very important biological information. And where is that in the brain? We don't know. I mean, there are studies of that. We know that in the ventral stream, there is response to biological motion. I don't have time to go over that. I highly recommend just look up biological motion literature. There is studies on that, but we don't know much, like where exactly in the brain that is happening, what are the brain structures that respond to biological motion, and how do they respond to that? Is it just biological motion or other motions also matter? Because there are these specific patterns of motion. Like if you drop a pencil on a solid table, it bounces with a very specific shape. And there is psychophysical evidence to show that we are decoding those motions. We know those motion motifs, even for non-living things. And where is it coded in the brain? How is it coded in the brain? That's the exciting question that take us to the last slide and your future, because you are the generation who is supposed to solve that problem. Thank you. I'm done. Questions?
So what you're basically hinting at is that like kind of like there's like some semantic knowledge that's like extracted from whatever the sound or like the visual information is, and it's something a lot more abstract that has meaning behind, you know, whatever sound it is, rather than the actual sound itself or the actual visual. Because it's what would that be? That would be mode. There's basically no particular mode, or basically no particular what is that? I don't know how to say the word, but it's like category in specific. It doesn't have to be vision or sound. That's really interesting.
Yeah, you know, the thing is that
amodal. Yes, thank you, Sophie.
Amodal, yeah. The thing is that we have been studying motion in the context of and there's no one to blame, actually. That's how you start studying something, in the context of simple vectors, right, that has speed and direction. And they are mostly built on top of linear systems background, V1 response to Gabor patches. So let's use sine waves moving. A lot of motion literature is using those kind of stimuli. And we are standing on the shoulders of those giants, actually, because they told us where in the brain are the main are the basic infrastructure for analyzing motion. However, I think we are way detached from the reality here. We have studied motion under very distilled conditions, right? And at the end of the day, the brain wants motion not to necessarily decipher like directions on a linear plane, on a flat plane. I need motion to understand what is out there, and I have a very opportunistic system that use all the cues, right? And it goes even beyond vision. It goes with the sound, and the connection with the sound is also pronounced in the brainstem, right? You have the superior caliculi. There are 4 of them. The top one, sorry, superior and inferior. You have the caliculi, and the top ones and the bottom ones are different. One pair of them are auditory, one pair of them are visual. And if you activate one of them visually, in an auditory way, the eyes will go. The other one will drive the eyes to look at there. If you have a motion signal, the same thing happens. It drives the eyes to look at that spot, like when a spark happens or a sound of a spark comes in. And I think, we've learned to study motion as a separate chapter in the book, right? When you study and take like whatever visual neuroscience or one of these classical textbooks, there is a chapter on motion, right? And there are motion labs that are studying only the motion thing, but the brain has not separated its areas based on the chapters in our textbooks, right? The motion is a very fundamental signal. It exists on the retina from the beginning, and we are constantly moving our eyes, and things are moving in the world. So and the visual system is an opportunistic system, right? So it has used it in multiple different parts in different ways. And while we know exciting things about it, we are far from done with that system, especially for these high-level semantic, more complex shapes of types of motion.
Yeah, I think it's a nice point. Like when you run if you want to find MT, you run a standard motion localizer, which is basically like random dots moving or random dots not moving. And that's very different from, you gave us the biological motion walking example, where that's a really specific stimulus. You could either have a walking stereogram, random dot stereogram, doesn't have to be stereo, I guess, the random dots walking, or the random dots doing something non-biologically meaningful. And that's what you would actually need to localize, something that you wouldn't get with a standard motion localizer, right? Like you asked earlier about MT hasn't grown, and I've been thinking about it since then. V1 hasn't expanded much either, right?
Right.
V1 is about as big as it used to be, and if MT is a primary area, it doesn't seem all that surprising that it would have been conserved the same way that V1 does. It does, as Fernando says, everything it needs to do, what MT has to do. But
comme si comme ça. I mean, V1 MT to V1 ratio has changed. Did it change? Let's go back to that plot.
Let's go back to that slide. It just seems as though the higher level motion behavior, like recognizing the gait of a particular person, let's say, not something that MT is doing, and it's not something you would expect V1 to do. You wouldn't expect V1 to recognize a particular person, a particular face, right?
And you're right, actually. You're right, Rosa. MT to V1 ratio has not changed.
Right.
Yeah.
So that for me is not that surprising. It's more that what are these higher order areas that doing with motion information, that is, this at the beginning I wasn't sure what you meant by higher order motion, because I know but now I'm like, oh yes, of course, biological motion is a great example of that. And that's not happening in MT. That's happening somewhere.
And exactly. And it's not just biological motion. It's a lot of things. Like you are wearing a cloth and the fabric waves in air, right? That's a very specific pattern of movement that is determined by the weight of the fabric, by the physics of the world, stuff like that. And we are sensitive to those things.
Right. It tells you, you can tell what that fabric would feel like, how heavy it would be, what it would be like to wave it around just by seeing that motion signal. Absolutely.
Exactly. And I am actually I feel guilty because there is lots of good literature on biological motion, and I don't have time to go in there, and that doesn't mean that it doesn't exist, but still there's lots of unknowns there for us. Even for faces, right? Faces are in the classical IT cortex. IT cortex is supposed to be like high on color, low on motion, right? Because color and motion are sort of opposite in a sense. But when you smile, when you move your eyes, how do we know how are those movements encoded in the face system? We know that the face neurons are sensitive to those. We know that. But how sensitive? How is it that it is encoded? Again, it's sort of a half-baked, half-done science. We know a little bit about that, but the big picture is far from clear.
Go ahead.
It's okay. It's a question that is related to the Gassenfar thing. So okay, we have these cells that are sensitive to kind of sound. And then if you show the gist of the mouth movement associated with that sound, these cells fire. So the question is, if you think of the McGurk illusion or the McGurk effect, in the end, you're using visual information to disambiguate the percept of the sound. So the question is then, are these cells tuned to any of these things at all? Or maybe they're tuned to something else, and these are just channels that inform those kind of states to which the system collapses when it has a percept?
Wow, that's another one of those good questions. That is such a big question, which is that makes it impossible to answer. Again, that's sort of the distinction between categorical response and tuning, right? So in one case, you have tuning of the cells, like in V1, they respond to that orientation. The response goes down, down, down as you change the orientation. That's tuning. But categorical response are these things that you see in IT, like it's response to faces. But there is no such parametric space to go from face to non-face objects, and you show that the tuning fades away. So we are kind of stuck with these because we don't know the parameters of that space, basically. So we are stuck with these categorical responses. And is it tuning or categorical response? We don't know, because we don't know how to morph a face to a chair, basically. And if you do that, is it systematically, is it encoding property of the object, which is systematically taking us from chair to face, or it's just responding to faces and ignoring chairs? We don't know. We don't know.
One other thing I wanted to just sort of ask about, and also just express frustration about, that I don't understand, is just like how temporal, like organizing information over temporal scales above like a flash stimulus presentation happens in neural circuitry. Like we have delay systems that we understand over a few neurons in a really small circuit, but how something like coherent motion of multiple points that inform you about a skeleton, like where on earth that would be happening? Like, do you need to build a structure first in order to be able to track that over time? I'm just thinking about this because like when I said that I see sort of object category information from like colorless, random dot, non-joint specific motion, I see it everywhere, and I don't know like how to explain mechanistically what these regions are
like
really
everywhere you can decode that.
I mean, like not literally everywhere, but yes, like in
it's
major,
very common.
In most ROIs that you would typically think to look, like even ventral as well. Like in the PFS, the performance is lower on the dynamic stimuli, but it's still decodable. And you also get cross decoding from like a picture of something to a video and back. You can decode.
Right. Yeah. Oh, wow. That's pretty amazing.
Yeah. So
from a still picture.
Yeah.
That's pretty impressive.
I mean, it would be nicer if my experiment was like beautifully parametric. They're like sort of they're not as beautifully controlled as they could be, but that the fact that that's possible is just very confusing to me. I have no idea how to mechanistically explain.
Yeah, right. I mean, whenever I see like good classification performance in the brain, the first question that comes to my mind is the task hard enough?
Right.
Basically, but if you have learning transfer from still image to the moving stimulus, to the dynamic stimulus, to the random dot stimulus, right? That's hard. That's hard enough, I guess. Yeah, and I don't know. I mean, I expect motion to be decoded everywhere in the brain, because it is a very fundamental property of the visual input. It's a dynamic input all the time. So it would be very narrow-minded to imagine only MT is dealing with motion, right? But what kind of motion is it that MT is dealing with? I don't think we have the clear picture. Like when you damage MT, your perception of motion goes away. Even that, I don't know to what extent goes away, right? When you do it in monkeys, the function comes back after some rewiring. They are bad for a few weeks or months maybe, and then it comes back. In humans, it's a mess. I mean, we have subjects, but many of those subjects have bigger lesions than just MT. Many of them are old, so the plasticity is not as pronounced in the brain. So even that notion of MT, if you destroy MT, motion is gone forever, I don't think data holds for that. I don't think so. I mean, MT is a tip of the iceberg, the first area that stands out because it's very motion selective. But I doubt other areas are, I mean, it's almost impossible not to use the motion signal.
Arash, remember the graphs that you showed us? I think it was on the retina, where if you showed the bar, if you flash the bar versus if you move the bar, you saw like the quick. So do you know if you see similar kinds of activity in MT when you do the same thing at MT to MT neurons?
Good question. Yes, you see that in MT. You see motion interpolation signal in MT as well. Yeah, which used to be surprising, but when you see that in retina, then MT becomes like boring.
Yeah, I think it's particularly interesting when you think of echinotopsia, because it's like it's almost kind of seems the way they describe their experience is that it's like everything happens in a flash, and it's not like you don't see this consistent motion. And when I think about it too, it seems like there is some sort of that there's some interaction going on in these two different concepts, because it almost seems as if like the coding of the like the fluent motion is lost, and I don't know, it just seems interesting. It seems like there's some sort of connection there.
Yeah, if you want
the way that the cell is behaving.
If you want to simulate a world without motion, or if you wanted to, I don't know, raise kittens without motion or something, you use a strobe light, right?
Yeah.
You create that flash experience where you've been in the club and they turn the strobe light on and you're moving your arms around, and you don't get the fluid motion.
Yeah. Right.
Yeah. You need that continuity. That I feel like that is what motion is the continuity of